Мгм. Спасибо. Good morning, dear students. So today we are going to discuss physiology of digestion. And our today's lecture topic is digestion in the oral cavity and digestion in the stomach. So the human and other animals must obtain their basic organic molecules from food, unlike plants, which can form organic molecules using inorganic compounds. In the other words, digestion is defined as the complex physiological process by which food that has entered the alimentary tract undergoes physical and chemical changes, and the nutrients in it are absorbed into the blood and lymph. Or another definition of digestion is that digestion is a complex physiological process by which food is broken down into simple chemical substances that can be absorbed and used as nutrients by the body. As for example, carbohydrates are broken down to monosaccharides, glucose. As example is glucose. Proteins are broken down to amino acids, and lipids are broken down to fatty acids and glycerol. There are 4 stages of digestion. The first stages of food processing, the stages of food processing. Ingestion, which is taken in nutrients. Digestion, using physical and chemical means to break down complex organic molecules into smaller usable parts. Absorption, pulling in digested molecules into the cells of the digestive tract, then into the blood. And egestion, the removal of waste food materials from the body. Egestion or elimination. According to the different features, the digestion can be classified into types. The first classification of the types of digestion is based on the origin of enzymes. And according to origin of enzymes, the types of digestion are following. Autolytic digestion, when food contains enzymes for digestion itself. Second type is so-called own digestion or proper digestion, when enzymes for digestion are produced by the organism. And the last one is so-called symbiotic digestion, when microorganisms of the large intestine take part in digestion. The other classification of the types of digestion is based on the localization of the process. So, according to localization, there are 2 types of digestion. Intracellular digestion, which includes phagocytosis, pino and endocytosis, and extracellular digestion, which also can be subdivided into distant digestion or cavital digestion. Cavital means in the cavities. The breakdown of food in cavity of intestine without direct contact with intestinal mucosa, far from place enzymes produced. It allows larger particles to be broken down into smaller particles and done by enzymes from pancreatic or intestinal juice. And second type is a contact digestion or membrane digestion, breakdown of nutrients on intestinal surface by enzymes bound to the enterocyte structures. Allows smaller particles to be broken down into simple monomers by enzymes lining the intestinal microvilli. Anatomically and functionally, the digestive system can be divided into a tubular part or a gastrointestinal tract or alimentary canal, so-called, and accessory organs and associated glands. The gastrointestinal tract or digestive system is a tubular structure extending from the mouth up to anus with a length of about 30 feet, which stays around 8 meters long. It opens to the external environment on both ends, and it is formed, GI tract, gastrointestinal tract, is formed by two types of organs, so-called primary digestive organs and accessory digestive organs. Primary digestive organs are the organs where actual digestion takes place. There are mouth and pharynx, esophagus, stomach, small and large intestine. And accessory digestive organs are those which help primary digestive organs in the process of digestion. There are teeth, tongue and salivary glands in the oral cavity, exocrine part of pancreas, which secretes pancreatic juice, liver and gallbladder, which also help in intestinal digestion. As we know, the oral cavity, teeth and tongue take part in mechanical processing, moistening, mixing food with saliva. Salivary glands will assist, and salivary glands secrete the lubricating fluid containing enzymes that break down carbohydrates primarily. Then pharynx, the muscular pharynx function is muscular propulsion of materials into the esophagus. The esophagus, as you know, transports materials to the next portion called stomach. The stomach is responsible for chemical breakdown of materials by acid and enzymes in gastric juice and mechanical processing through the muscular contraction. Then small intestine is responsible for enzymatic digestion and absorption of water, organic substrates, vitamins, ions. The digestion in the small intestine depends upon the secretion of pancreas. Exocrine cells secrete buffers and digestive enzymes. And the endocrine cells of pancreas, as you know, secrete hormones. The hormones which are so-called GIT, gastrointestinal hormones. Intestinal digestion also associated with the functions of liver and gallbladder. The main function of the liver, as you know, secretion of bile, which is very important for the lipid digestion. And has also many other vital functions we'll discuss on the next lecture. And gallbladder, which stores and concentrates bile. And finally, large intestine, which is responsible for dehydration and compaction of undigestible materials in preparation for their elimination. The general organization of gastrointestinal tract. So, a typical section of the intestinal wall showing the following layers from the inner to the outer surface. So, the first layer is mucosa. The mucosa consists of the epithelium, which usually simple columnar with goblets, lamina propria, connective tissue deep to epithelium, and muscularis mucosa, which produces folds in the small intestine or rugae in the stomach. The next layer is submucosa. The submucosa is made up of loose connective tissue and contains submucosal plexus and blood vessel. The submucosal plexus, also called Meissner's plexus. Muscularis externa consists of smooth muscles, usually 2 layers, controlled by the so-called Auerbach's or mesenteric plexus. There are outer longitudinal muscle layer and inner layer of circular muscle. And the last one, serosal layer of mesentery or adventitia, depending on location. In addition, a sparse layer of smooth muscle fibers, the muscularis mucosa, as I said already, lies in the deep layers of the mucosa. The individual smooth muscle fibers in the gastrointestinal tract are arranged in bundles of as many as 1,000 parallel fibers. In the longitudinal muscle layer, these bundles extend longitudinally down the intestinal tract. In the circular muscle layer, they extend around the gut. Within each bundle of muscle fibers are electrically connected with each other through large numbers of gap junctions. Therefore, electrical signals can travel from one to the next fiber. Same time, the bundles fuse with each other at many points. Therefore, each muscle layer functions as a syncytium. When an AP is elicited anywhere within the muscle mass, it travels in all directions in the muscle. Although a few connections exist between the longitudinal and circular muscle layers. That excitation of one of these layers usually excites the other as well. The smooth muscle of the GIT undergoes almost continual slow electrical activity. This activity tends to have 2 basic types of electrical waves, so-called slow waves and spikes. These terms are well known to you from the physiology of excitable tissues. The smooth muscle of the gastrointestinal tract has spontaneous, rhythmical fluctuations in membrane potential between about -65 and -45 millivolts. This is called basic electrical rhythm. Abbreviature is BER. It is initiated by the interstitial cells of Cajal. stellate mesenchymal pacemaker cells with smooth muscle-like features that send long multiply branched processes into the intestinal smooth muscle. The basic electrical rhythm itself rarely causes muscle contraction. However, they do control the appearance of intermittent spike potentials, and the spike potentials in turn actually cause the muscle contraction. The intensity of slow waves varies between 5 and 15 millivolts, and their frequency ranges in different parts of the gastrointestinal tract between 3 and 12 per minute. About 3 in the body of the stomach, as much as 12 in the duodenum, and about 8 or 9 waves in the terminal ileum. Therefore, the rhythm of contraction of the body of the stomach usually is about 3 per minute, of the duodenum about 12 per minute, and the ileum 8 to 9 per minute. In the colon, basic electrical rhythm rate rises from about 9 per minute, at the cecum to about 16 The spike potentials are true action potentials. They occur automatically when the resting membrane potential of the gastrointestinal tract smooth muscle becomes more positive than about negative 40 millivolts. The normal resting membrane potential is in between 50 to 60 millivolts. The spike potentials appear on the peaks of slow wave potential, and the higher the slow wave potential rises above this level, the greater the frequency of the spike potentials, ranging between 1 to 10 spikes per second. The depolarizing portion of each spike is due to calcium ions influx, and the repolarizing portion is due to potassium ions efflux. Many factors affect the BER, or basic electrical rhythm. These factors are stretching of the muscle, stimulation by acetylcholine, stimulation by the parasympathetic nerves that secrete acetylcholine, stimulation by gastrointestinal hormones. Factors that make the membrane potential more negative or that hyperpolarize membrane will cause hyperpolarization of membrane are epinephrine and norepinephrine secreted by postganglionic sympathetic fibers, and hormones of adrenal medulla, epinephrine and norepinephrine. The functions of basic electrical rhythm is to coordinate peristaltic and other motor activity. Contraction occurs during the depolarizing part of the wave, after vagotomy or transection of the stomach wall. Peristalsis in the stomach becomes irregular and chaotic. Functions of gastrointestinal tract. The functions of gastrointestinal tract can be also classified as digestive functions and non-digestive functions. So look, the digestive functions of gastrointestinal tract are secretory function, motor function, absorption, and excretory function. All these following function as a defensive function, homeostatic function, and regulatory function, regulatory or endocrine function, are so-called non-digestive functions, but which are also very important for the normal process of digestion in the GIT. Now we'll discuss briefly the general principles of GIT functions control. So, there are two mechanisms of regulation. As you know it well, neural mechanism and humoral mechanism. The neural control of the gut wall showing the myenteric and submucosal plexuses, so-called enteric nervous system, which is an intrinsic controlling mechanism. We also know this system, enteric nervous system or metasympathetic nervous system. It's one of the variants of metasympathetic nervous system activity. Extrinsic control of these plexuses is due to functions of autonomic nervous system, its sympathetic and parasympathetic divisions. And the hormonal control is based on the secretion of gastrointestinal hormones by APUD system over GIT. As you know, the local neural control based on intrinsic nerve plexuses of metasympathetic nervous system. This intrinsic system of GIT extends from esophagus up to the anus, and it is divided into myenteric or Auerbach's plexus, which lies between longitudinal and circular muscle layer. And the main function of this plexus is control over GIT movements, the control of motor activity of gastrointestinal tract. And the second division is a submucosal plexus or Meissner's plexus. It lies in submucosa and its function control GIT secretion and local blood flow. Subfunctions, it serves sensory functions by receiving signals from GIT epithelium and from stretch receptors of GIT wall. So, collectively, these neurons constitute so-called enteric nervous system, as I said already, which contains about 100 million sensory, inter and motor neurons. It is connected to the CNS by the parasympathetic and sympathetic fibers, but can function autonomously without these connections. The myenteric plexus innervates the longitudinal and circular smooth muscle layers, as I said already. and which is concerned primarily with motor control, whereas the submucous plexus innervates the glandular epithelium, intestinal endocrine cells, and submucosal blood vessels, and is primarily involved in the control of intestinal secretion. The neurotransmitters of enteric nervous system include acetylcholine, norepinephrine, serotonin, GABA, ATP, nitric oxide, and other peptides and polypeptides. The autonomic nervous system controls the activity of enteric nervous system by sympathetic and parasympathetic fibers. The parasympathetic nervous system's activity depends upon the vagus nerve that innervates GIT from esophagus up to proximal 2/3 of transverse colon. Also, pelvic parasympathetic nerve S2 to S4 innervates GIT from distal 1/3 of transverse colon. Its function to increase peristalsis and tone, relaxes sphincters, and to increase digestive secretion. Sympathetic nervous system. Preganglionic fibers, the red color as you see, the blue color parasympathetic, the red color sympathetic. Preganglionic sympathetic fibers originate in T5 to L2 segments of spinal cord, pass through sympathetic chain, synapse with postganglionic neurons in celiac and mesenteric and hypogastric ganglia, and postganglionic fibers innervate GIT, gastrointestinal tract. Its functions: to decrease peristalsis and tone, contract sphincters, and decrease digestive secretion. So here on that slide you may see that GIT receives a dual extrinsic innervation from the autonomic nervous system with parasympathetic cholinergic activity, generally increasing the activity of GIT and increases secretion of GIT, and sympathetic noradrenergic activity, generally decreasing it while causing sphincters to contract. And the last one is the hormonal regulation of GIT, another one mechanism. The pericrine effect of chemical messengers and gastrointestinal hormones is the basis of this mechanism of regulation called humoral regulation. The hormones and humoral agents secreted by cells in the mucosa and transported in the circulation to influence the functions of the stomach, the intestine, the pancreas, and the gallbladder. They also act in a pericrine fashion. So, as you see, there are two mechanisms of GIT functions control. These mechanisms are neural mechanism and humoral mechanism, which can perform their functions at two levels, local and central levels. Now we are going to discuss the functions of the oral cavity. As you know, in the mouth, food is mixed with saliva and then propelled into esophagus. And there are two basic processes that take place in the oral cavity. First process called mechanical process. Mastication or chewing breaks up large food particles, mixes the food with the secretion of the salivary glands, and rolls it into a moist, soft mass called bolus, suitable for swallowing. So, the functions of the oral cavity is mechanical breakdown of food, then chemical breakdown of food. The chemical breakdown of food is based on the action of salivary enzymes. Then, propelling food into the next portion of the GIT by swallowing. There are also analysis of taste due to the series of taste receptors of the oral cavity. There is also protective function, and moistening of tongues and lips for articulation. Saliva. The secretion of saliva ranges between 0.8 to 1.5 liters per day. It is a slightly acid solution, 6 to 7.4, which contains salts and organic substances secreted mainly by 3 pairs of salivary glands. These glands are sublingual, submandibular, and parotid glands. The functions of saliva is a chemical processing of food due to enzymes. As you know, saliva contains salivary amylase, alpha amylase, which is also called ptyalin, which splits starch into maltose and alpha-limit dextrins. It also contains weak lingual lipase secreted by glands on the tongue, which plays a minor role in the fat's digestion, as you understand. It also contains mucin glycoprotein, which lubricates the food and protects the oral mucosa. It also contains lysozyme that attacks the walls of bacteria. It contains lactoferrin, which binds iron and has bacteriostatic effect. And it also contains minerals, sodium, potassium, chloride, and bicarbonates. The functions of saliva are following: chemical processing of food, it moistens food and facilitates swallowing. In the process of moistening food, chemical and mechanical breakdown due to chewing movement, due to mastication, the food becomes a bolus. It lubricates tongue and lips and facilitates articulation, as I said already. It has protective function due to the biological active substances it contains listed earlier. And there is also excretory function. Some substances can be excreted through the saliva. This figure, what it illustrates? It illustrates the parasympathetic nervous pathways for regulation of salivation, showing that parasympathetic, as you see, showing that salivary glands are controlled mainly by parasympathetic signals from salivary nuclei that are located in the medulla oblongata and pons, as you know, and are excited by both taste and tactile stimuli from the tongue and the other areas of the mouth, from the receptors of the oral cavity. Some certain tactile stimuli, such as presence of smooth objects in the mouth, like a pebble, for instance, cause marked salivation, whereas rough objects cause less or even inhibit salivation. Salivation can also be stimulated or inhibited by impulses arriving in salivary nuclei from higher centers of the central nervous system of the CNS. When the person eats or smells favorite food, salivation is greater than when disliked food is smelled or eaten. It is clear. The appetite area of the brain, which is located in the anterior hypothalamus, taste and smell areas of cerebral cortex and amygdala also regulate these effects. Sympathetic stimulation can also increase secretion of saliva, but much less than parasympathetic. So, then does parasympathetic secretion has? The sympathetic nerves originate from the superior cervical ganglia and travel along the blood vessels to the salivary glands. And it also, sympathetic nervous system, also affects secretion, not only secretion, but also blood supply of the glands. Phases of salivary secretion. So, salivary secretion is based on neural and neural control, which in turn can be divided into unconditioned and conditioned. Unconditioned and conditioned. The unconditioned secretion of saliva takes place when food is taken into the mouth, which in turn stimulates receptors in the oral cavity, chemo and mechanoreceptors. The food stimulates nerve endings, as I said, receptors in the mouth, and a stream of ingoing nerve impulses will travel along taste nerve to salivary nuclei in the medulla oblongata and pons. The result of this is that outgoing nerve impulses of secretomotor nerves from the autonomic nervous system to gland evoke secretion of saliva. Another mechanism of salivation based on conditioned reflex. For example, sight of food, smell of food, even thought of food and sounds of its preparation become associated with the taste of food. And a result of this, that ingoing nerve impulses from, for example, visual receptors or olfactory receptors, will travel to the corresponding areas in the CNS, in the brain cortex, for example, to the visual center. The same time, simultaneously, ingoing nerve impulses will travel to the salivary center from the taste receptors of the mouth. And the result of this is association pathways which become established between two centers in the brain. One center which is responsible for conditioned secretion of saliva, for example, visual center, and the unconditioned center of salivation. The parasympathetic nerve releases acetylcholine, which greatly increases salivary secretion, and it also releases vasoactive intestinal polypeptide, VIP, which dilates the salivary gland blood vessels, and by that way it increases salivation. The sympathetic nerves will cause secretion of small amounts of saliva rich in protein and glycoprotein. And also, for addition, that the parotid glands secrete the serous type watery saliva. The submandibular and sublingual glands secrete both the serous type and mucus. The buccal glands secrete only mucus saliva. Mastication, as I said already, or chewing, is when the food is broken down, mixes with the saliva, and rolling food into a moist, soft mass, which is called bolus. Most of the muscles of chewing are innervated by the motor branch of the CN5 trigeminal nerve, and the chewing process is controlled by nuclei in the brain stem. Chewing of food is important for digestion of all types of food, and it aids in the digestion of food for the following reasons. The digestive enzymes act only on the surface of food particles, so it increases the surface area for contact with digestive enzymes. And it also grinding the food to a very fine particles. Swallowing. The swallowing is the complex act which is initiated voluntarily and completed involuntarily. This process is staged process, and it can be subdivided into 3 stages. The first stage of swallowing, called oral stage, it is voluntarily and it initiates the swallowing process. Second stage of swallowing, called pharyngeal stage. This stage is involuntary, and it constitutes the passage of food through the pharynx into the esophagus. And the last one, third stage of swallowing or deglutition, called esophageal stage, which is a slow, involuntary. It promotes passage of food from the pharynx to the stomach. The reflex response is triggered by afferent impulses in the trigeminal nerve, glossopharyngeal nerve, and vagus nerve. These impulses are integrated in the nucleus tractus solitarius and nucleus retroambiguous in the medulla oblongata, and the efferent fibers from the pharyngeal will pass to the pharyngeal musculature and the tongue via CN5, 10, 11, and 12, and it will promote deglutition or swallowing. Now we should discuss briefly the digestion in the stomach, which follows digestion in the oral cavity. So, the peristalsis of the esophagus pushes swallowed food from one end of the esophagus to the other circular and due to the circular and longitudinal muscle contraction towards the lower esophageal or gastroesophageal sphincter. The lower esophageal sphincter remains closed until food is pushed through it by peristalsis into the stomach. By the way, the lower esophageal sphincter is not a true sphincter muscle that can be identified histologically. The stomach. The stomach is a G-shaped bulge or pouch. Structurally, stomach can be divided into the cardiac region or cardia, fundus, greater and lesser curvature, body of stomach that comprises 2/3 of the stomach, and pyloric region or antrum, the distal portion of the stomach. The outer surface, the outer wall of the stomach consists of 3 smooth muscle coats, 3 smooth muscle layers, as you may see, longitudinal, circular, and oblique muscles. The inner surface of the stomach is thrown into a series of folds known as rugae, which increase the surface area of gastric mucosa and very important for the digestion in the stomach. The opening of these folds into the stomach lumen called gastric pits. The mucosal cells lining them are called exocrine gastric glands. Exocrine gastric glands. There are several types of cells in the gastric glands that collectively secrete secretion commonly called gastric juice, 2 to 3 liters per day. Goblet cells or mucus cells secrete mucus that protects mucosa from acid and from pepsin also. The parietal cells or oxyntic cells, parietal cells between a mucus cells and a chief cells, secrete hydrochloric acid and intrinsic factor. Hydrochloric acid kills bacteria. The intrinsic factor binds with vitamin B12 and allows absorption in ileum. Vitamin B12 is required for maturation of red blood cells in the bone marrow. A patient with gastrectomy has to receive B12 orally together with intrinsic factor or through injections to prevent the development of pernicious anemia. Chief cells. The chief cells, which contain zymogen granules and secrete pepsinogen, which is an inactive form of the protein digestive enzyme pepsin, activated by hydrochloric acid. When it is activated, it becomes pepsin, and it starts chemical breakdown of protein to small peptides. The chief cells also secrete gastric lipase, which digest emulsificated fats, and as well as a lingual lipase plays a minor role in fats digestion. G cells in the antrum. G cells. The G cells, look here, yeah, the G cells secrete gastrin, gastrointestinal hormone, which stimulates growth of gastric glands, and it stimulates secretion of large amounts of gastric juice. There are also other endocrine cells, as for example, histamine secreting cells, which act as paracrine cells controlling acid secretion in adjacent parietal cells. The surface epithelium cells secrete bicarbonates into a covering and resistant layer containing mucus and making it alkaline. As a result, the epithelial surface remains close to neutral. Gastric juice also contains minerals, ions, sodium, potassium, chloride, phosphate, magnesium. It also contains mucin, lysozyme, urea, and other organic and inorganic substances. The gastric mucosa contains many deep glands, which also can be identified by their topography. The pyloric zone. The pyloric glands mostly secrete mucus and gastrin. In the cardiac zone, the cardiac glands, so-called cardiac glands, secrete mostly mucus. And finally, fundy glands. The fundy glands secrete, or gastric glands also so-called, they contain all types of secretory cells, parietal cells, chief cells, and goblet cells. Here on that slide, you may see the composition of gastric juice. And its pH, as you see, it fluctuates and ranges in between 1.6 to 3.2. It contains pepsin, protein digestive enzyme. It contains weak gastric lipase, playing a minor role in digestion of fats. It also contains other components. We have listed them earlier. Here on that slide, you also may see the localization of these glands, cardiac glands, gastric glands, the glands in pylorus or pyloric glands, and the type of secretion by each type of glands. Only gastric glands contain all types of cells. Functions of the hydrochloric acid. pepsinogen activation. It activates pepsinogen, which then becomes active pepsin. Then it maintains an optimal pH. It denaturates proteins, takes part in swelling of proteins. It also has bacteriostatic effect. It regulates the gastric emptying and stimulates production of gastrin by G cells and activates duodenal enterokinase. Functions of the stomach. The first function of the stomach is a reservoir for food during meals. It is clear. Next function is secretory function. It is based on the secretion of gastric juice, composition of which we discussed earlier. The motor function. The motor function of the stomach is based on different types of motor activity. It is, simply speak, a mixing chamber. This mixing chamber which churns food into semi-fluid chyme, it is called chyme, and then delivers it into the next portion of gastrointestinal tract called duodenum. Next function, absorption. Absorption of water, alcohol, some drugs takes place in the stomach. There is also defensive function. Defensive function, we discussed it already earlier. Regulatory function, regulatory or incretory function, secretion of gastrointestinal hormones. Hemopoietic function, which is related to secretion of intrinsic factor or causal factor. And excretory function, urea, uric acid, creatinine, leads, and some drugs also are excreted. And the generally homeostatic function. Homeostatic function, it plays a very important role in the regulation of pH through the transportation of hydrogen and bicarbonates. Phases of gastric secretion. So, regulation of gastric secretion and gastric motility. Gastric motility and gastric secretion are regulated by neural and humoral mechanisms. The neural components are local autonomic reflexes involving cholinergic neurons and impulses from the CNS by way of the vagus nerves. The humoral components are the gastrointestinal hormones. Phases of gastric secretion are subdivided into following phases. The first phase called cephalic phase. The cephalic phase, as well as a cephalic phase of salivation, are divided into conditioned and unconditioned. The presence of food in the stomach, the presence of food in the mouth reflexly stimulates gastric secretion. The efferent fibers for this reflex are in the vagus nerves. Vagally mediated increases in gastric secretion are easily conditioned. The sight of food, smell, and thought of food increase gastric secretion. These increases are due to alimentary conditioned reflexes that become established early in the life. The cephalic influence are responsible for 1/5 to 1/2 of the gastric juice secreted in response to a meal. So, conditioned and unconditioned. Next phase called gastric phase. Food in the stomach accelerates the increase in gastric secretion produced by the sight, smell, and presence of food in the mouth in the previous phase. Receptors in the wall of the stomach and mucosa respond to stretch and chemical stimuli, and it excites the long vagal reflexes and local enteric reflexes, and also gastrin mechanism, which ensures continued gastric juice secretion and also ensures its motility. So this phase, gastric phase, is responsible for 2/3 of total gastric secretion. The last phase of gastric secretion, called intestinal phase. This phase starts, look here, this slide, cephalic, gastric, and intestinal. It starts when chyme, which is semi-fluid food following digestion in the stomach, distends the duodenum, both vagus and intrinsic nerve plexus, duodenal mucosa, which secretes secretin, CCK, GIP, entering the bloodstream, inhibits gastrin release and secretion of the acid gastric juice, and also inhibits gastric motility. Gastrin secretion is inhibited when the pH of gastric content falls to 2. It also inhibited by fear, anger, anxiety, and sympathetic nerve stimulation. Here on that slide you may see the mechanisms of each phase: cephalic phase, conditioned and unconditioned, conditioned and unconditioned, gastric phase, and finally intestinal phase. Types of gastric motility. So, the motor functions of the stomach are following. First function is a storage of large quantities of food, because, as we said, the first function of the stomach is so-called reservoir function. And mixing of food with gastric juice until it forms semi-fluid mixture called chyme. And also slow emptying of food from the stomach into the small intestine. The types of stomach's motility. An intense contraction are seen in the empty stomach, when it is empty for a long time. These are rhythmic peristaltic contractions in the body of the stomach. When it occurs in the stomach, the person feels a sensation of pain. They begin 12 to 24 hours after the last meal and reach greatest intensity in 3 to 4 days of starvation. Hunger contractions are often associated with a feeling of hunger. When they become extremely strong, they fuse together to cause a continuing tetanic contraction lasting for 2 to 3 minutes. When food enters the stomach, a vagal reflex greatly reduces the tone of the muscular wall of the body, so it can bulge outward, accommodating greater quantities of food, up to a limit of about 1.5 liters. This reflex is triggered by movement of pharynx and esophagus and enteric nervous system also, that releases inhibitory neurotransmitters, and it also vagally mediated. The mediator adenosine. Peristalsis occurs while food is in the stomach. Peristalsis originates from the pacemaker zone on the greater curvature of the stomach. Food moves toward the antrum and mixes with gastric juice. The semi-fluid mixture called chyme is formed. Chyme will be carried through the pylorus in small portions, in small quantities. The sphincter then contracts, forcing chyme back by retropulsions, and mixes the food with gastric juice again. Antral systole. The antral systole is the contraction of distal stomach caused by each wave of peristalsis. It also helps in gastric emptying. Antiperistalsis, as you know, can be seen in a complex reflex act coordinated by medulla oblongata and called vomiting. And the final stage is gastric emptying. Gastric emptying. The motility of the stomach and the stomach emptying depends upon different factors. So, the factors that promote motility of the stomach and that promotes gastric emptying are gastric distension, gastrin, acetylcholine, motilin, and increased vagus tone. The factors that inhibit motility of the stomach and its emptying or decrease gastric emptying are following: CCK, GIP, and gastrin, which are gastrointestinal hormones, have a mild effect. Then the degree of duodenal distension, then proteins, fats, the products of proteins and fats, digestion in the chyme, high acidity of the chyme and high osmolarity of the chyme, and also decreased vagal or increased sympathetic stimulation. So these factors will inhibit gastric emptying. Methods of gastric secretory function examination. Basically, all methods of gastric secretion and gastric motility investigation are subdivided into experimental method. Here you may see simple fistula method, or simple fistula of the stomach by Basov, experiment of sham feeding with esophagotomia by Pavlov, isolated miniature stomach or pouch by Pavlov. So these methods are so-called experimental methods. And clinical methods, yeah, using probe, endoscopy with biopsy, intragastric pH metry, without probes, electrogastrography, X-ray method, X-ray examination, the radio peel, laboratory methods. Here on that slide, you may see some procedure of sham feeding. For example, here you see the sham feeding on this picture. The simple fistula, the hollow tube inserted into the lumen of the stomach. And through this tube, the gastric juice can be collected and investigated. The influence of different factors on its secretion and its composition also can be examined. And this one, a hole, as you see, is made in the neck of an anesthetized dog. Esophagus is transversely cut and the cut ends are drawn out through the hole in the neck. When the dog eats food, it comes out through the cut end of esophagus. And but the dog has the satisfaction of eating the food. That's why this experiment called sham feeding. This experimental procedure is supported by the preparation of Pavlov pouch, which with a fistula from the stomach. The fistula opens to exterior and used to absorb the gastric secretion. Sham feeding is useful to demonstrate the secretion of gastric juice during cephalic phase. In the same animal after vagotomy, sham feeding does not induce gastric secretion. And this experiment proves the role of vagus nerve during cephalic phase. And here you also may see the Pavlov's pouch and Heidenhain pouch. Methods of gastric motor functions examination are electrogastrography, the recording of the electrical activity, and gastrography. So today on the lecture we have discussed the general principles of GIT regulation. We have discussed the types of digestion, stages of digestion, and some aspects of digestion in the two divisions of the GIT, in the oral cavity, digestion in the oral cavity, secretion, motility, absorption, regulation of secretion and motility in the oral cavity, and the same processes in the stomach, secretion, motility, absorption. So on the next lecture, we are going to discuss the next portions of gastrointestinal tract and basic principles of their organization and their function. So thank you for your attention. See you on the next lecture. Goodbye. Давайте уточним.